All right, we are being joined right now by Dr. Lena Smirnova, an assistant professor at Johns Hopkins University in Baltimore. Welcome to Music City. Thanks for sitting down with us today.
Thank you for inviting me.
So, why is there a push right now to consider environmental factors leading to autism?
There is a lot of things to think about when we think about the autism, and why we think about environment per se is also because we're seeing huge increase in autism cases over the last decade, which cannot be explained only by better diagnostics, for example, or better awareness of general public, or for example, genetics, right? So there is certain component which is genetic, but we are looking for something else which is outside the genetic spectrum, and that's an environment. And environment, it's not only exposure per se, it's also lifestyle, infectious diseases, mother smoking, for example. It's everything considered to be environment. So, and we're looking for clues there as well.
And how could that interaction of genetic and environmental factors influence autism?
So autism is very heterogeneous disease, so it's a spectrum, right? So that's why we have different individuals on different spectrum of the disorders. And it's also multigenic disease. So there's a lot of genes which are associated with autism, but every individual gene doesn't necessarily lead to more than 1% of the cases. And also, if individuals with the same mutation, they can have completely different phenotypes, or they have completely different symptomatic. So and that's where we see that environment can play a role and put the people with the same mutation on different spectrum, or aggravate them, for example, the symptomatic.
Let's talk about your specific research. As I mentioned, you're an assistant professor at one of the best universities in the entire country. Tell me a little bit about your specific research and how you are planning to sort of expand that moving forward in the years to come.
In my research, I'm using induced pluripotent stem cells derived model. So this is the new advanced technologies. We call them brain organoids. So it's three-dimensional cultures of brain cells derived from stem cells. So since it's from induced pluripotent stem cells, I can have different genetic background, autistic versus typically developed. And then when I differentiate them in those small tiny balls of the cells, which we call brain organoids, I can see how certain environment influence the differentiation, and whether, for example, those which have a mutation in an autism risk gene are more sensitive to the environmental exposure. And this, for example, we published as a first proof of principle study last year in Environmental Health Perspective journal, where we looked into the high-risk autism gene called CHD8, which is regulate transcription, and a chemical which is under radar for many years, which is organophosphate pesticides, chlorpyrifos. And we looked how these chlorpyrifos exposure synergize with CHD8. And we saw some synergy in certain molecular pathways. So we saw that the exposure will make the effects of mutation worse. And how we are moving forward, we are currently working, we have also grants from FDA and EPA to look at different other exposures. For example, with help from FDA, we look into the metal mixtures. Heavy metals like lead, arsenic, cadmium, chromium, they're all around us, and in some places, kids are exposed, for example, or pregnant women are exposed to higher levels of those.
That's very impressive, and best of luck to you on your
Thank you.
research moving forward. Dr. Lena Smirnova, an assistant professor at Johns Hopkins University. Enjoying the conference, and thanks for joining us.
Thank you, I will definitely do that. Thanks.